Magandan
Maganan
Ravi po, myself, who is this?
Maganan gabi po. Ako si Raj.
Magandan Ravi po, myself, Daniel Trivedi. So today we are going to explain our presentation on apoptosis and necrosis. So with this, I will start. Sure. So let's start. Today we'll present a presentation on apoptosis and necrosis. Yeah. Let me first fix this. Yeah, let's go. So the first slide begins with, well then let's get started. Apoptosis. It is also called programmed cell death, and definition of it is apoptosis is a regulated, controlled process of cell death, often referred to as programmed cell death. It eliminates unnecessary or damaged cells in a structured and clean manner. Mechanism. It's triggered by internal or external signals. Apoptosis activates enzymes, example caspases, that break down the cell from within. Characteristics. The cell shrinks, its DNA condenses, and it forms blebs that are eventually cleared by nearby cells or immune cells. There is no damage to surrounding tissue or inflammation. Causes. It can be triggered by DNA damage, cell stress, or external signals like immune system activation. Outcome. The process is clean and non-inflammatory, preventing harm to surrounding tissues. Example. Removal of excess cells during development or elimination of infected or cancerous cells. So this is the flow chart on how the apoptosis actually occurs. So let's discuss the apoptosis flow chart, which highlights the key pathways and steps involved in programmed cell death. Apoptosis is essential for maintaining cellular balance and removing damaged or unwanted cells. So we start at the initiator stage, where apoptosis can be triggered through two distinct pathways. The extrinsic pathway. This pathway is initiated by external signals. These signals come from death ligands like FASL, or TNF alpha, which bind to death receptors on the cell surface. Once activated, these receptors recruit and activate caspase 8 and initiator caspase. Caspase 8 then activates the executional caspase 3, and caspase 7, which drive the final steps of apoptosis. The intrinsic pathway. This pathway is activated by internal stress such as DNA damage, oxidative stress, or loss of survival signals. The stress signals cause the release of cytochrome C from the mitochondria into the cytoplasm. Cytochrome C binds with APF1, apoptotic protease activating factor 1, forming the apoptosome complex. This complex activates caspase 9, another initiator caspase. Caspase 9, like caspase 8, activates caspase 3 and caspase 7, leading to the execution phase. Now, let's move on to the execution phase. The activation of caspase 3 and caspase 7 leads to several morphological and biochemical changes in the cell, including cell shrinkage, where the cell condenses and reduces in size, nuclear fragmentation, breaking down the nucleus into smaller pieces, membrane blebbing, where the cell membrane forms small bubble-like protrusions. These changes result in the orderly dismantling of the cell, ending with its removal by phagocytes without causing inflammation or damage to surrounding tissues. So, in summary, the flowchart provides a clear representation of how apoptosis is tightly regulated, ensuring controlled cell death through either external or internal triggers. The process culminates in the efficient removal of cells critical for tissue health and homeostasis. Now I will call my friend Raj to explain the summary of the pathway. My part is summary of the pathway.
Okay. Apoptosis. Initiator. Extrinsic pathway. Triggered by death receptor. Activate cas which leads to caspase 3 by 7 activation. Intrinsic pathway triggered by the cellular stress. Example like DNA damage. Cytochrome C release activates APAF1, leading to caspase 9 activation and eventually activates the caspase 3 by 7. Execution. Caspase 3 by 7 drives the execution phase by including cell shrinkage, nuclear fragmentation, membrane blebbing, resulting in cell death. Necrosis. An uncontrolled cell death. Definition. Necrosis is an uncontrolled pathological form of cell death caused by factors like trauma, toxins, or lack of oxygen. Mechanism. It occurs due to the acute damage resulting in swelling of the cell, rupture of the cell membrane, and release of content into the surrounding tissue. Characteristics are Cells, cells swell and burst, causing inflammation and damage to the surrounding tissues. Common causes includes injury, infection, or loss of blood supply. Ischemia. Outcomes are necrosis leads to inflammation and tissue damage, which can spread to nearby cells. Example like myocardial infarction, heart attack, stroke, or gangrene. Now I call my friend there to explain further.
So thank you, Raj. So now I will explain the necrosis flowchart. So let's go through the necrosis flowchart, which breaks down the different forms of cell death under the broader category of necrosis. The necrosis can occur in both regulated and unregulated forms, and this diagram explains each. So first, necroptosis. Necroptosis is a regulated form of necrosis initiated by external signals, particularly death receptors like TNFR or FAS. Once these receptors are activated, key mediators including RIPK1 and RIPK3 are recruited. These in turn activate MLKL, a protein that forms pores in the cell membrane. The formation of these pores causes cell rupture, which results in the release of cellular contents and inflammation. Second, pyroptosis. The pyroptosis is another form of regulated necrosis triggered by the activation of inflammasomes, example NLRP3/ASC. This inflammasomes activate caspase 1, which then cleaves gasdermine D. The gasdermine D plays a critical role in this process by forming pores in the cell membrane. This pore formation leads to cell rupture and the release of pro-inflammatory contents, driving an inflammatory response. Third, ferroptosis. The ferroptosis is caused by ion overload, GPX4 inhibition, or oxidative stress, which results in excessive lipid peroxidation. The lipid peroxidation damages the integrity of cell membranes, ultimately leading to membrane rupture and cell death. This type of necrosis is highly dependent on oxidative damage and the iron metabolism. The fourth, unregulated necrosis. Unlike the regulated forms, unregulated necrosis occurs due to acute injuries such as trauma, ischemia, or exposure to toxins. This injury results in cell membrane damage and energy depletion. This execution phase involves cell swelling, rupture, and a subsequent inflammation, which can cause significant tissue damage. So that's all from this flowchart. Seconds, another slide. The necrosis summary. So the necroptosis initiators are triggered by death receptors such as TNRF death path, as explained earlier in the flowchart. The key mediators RIPK1, RIPK3, and MLKL. Execution MLKL forms membrane pores leading to cell rupture. The second, pyroptosis. Initiators are triggered by inflammasomes, example LLRP3/ASC and caspase 1 activation. The mediator are gasdermine D. Execution. The gasdermine D forms pores in the membrane, which leading to the cell rupture and inflammation. Third, ferroptosis. The initiators are caused by ion overload, GPX4 inhibition, or oxidative stress. Execution leads to lipid peroxidation and membrane rupture. Fourth, unregulated necrosis. The initiators are triggered by acute injury, example trauma, insomnia, toxins, or cell membrane damage. The execution results in swelling, rupture, and inflammation. So thank you for that's all from our side. Thank you
Thank
for
you for
Salamat
that's all
bro.
from our side. Thank you. Salamat. Salam.